Excellent. Okay, so we're live and I already see a few people dialing in. Good afternoon, everyone, and welcome to our November Journal Club. We have a nice group assembled for today, so as folks are still dialing in, we'll just let it run for a minute or two before we officially get started. So thank you for being here, and we'll dive in a minute. Fantastic. So we're 2 minutes after the hour. So let's go ahead and get started to be respectful of everyone's time. Welcome everyone to our November Journal Club, our monthly Ask Me Anything discussion about some of the sort of most cutting edge, the most important publications on digital medicine, sort of fresh to the academic literature. I am delighted to welcome Vaibhav today, who is going to walk us through a really terrific and recent publication on using smartphones and wearable devices to monitor behavioral changes during COVID-19. Vaibhav, I can't imagine a more pertinent topic. We've got a great crowd on the line today for discussion. Let me just pause for a second before I hand the reins over to you and let folks on the line know how they can participate in this discussion. So quick housekeeping note, we are recording today's discussion. We will always disclose that. The good news there is it does mean we'll be able to post the recording and the slides from today's presentation to the DIME website a little bit later today. Also, for folks who haven't attended a journal club before, Vaibhav will walk us through some of the high points from the publication, from the author's perspective, really sort of tease out some of the most interesting pieces of the publication, put them into his own voice, and then we will throw it open for conversation. There's a number of different ways that you can engage. You can raise your hand, not in person, but actually in the participant list. I'll be keeping an eye out. If you're somewhere where you can chat, I will unmute you and ask you to pose your question directly. If you would prefer to type your question into the chat or Q&A, I will be moderating those too. So I look forward to a terrific discussion. This is a really important topic. Vaibhav, welcome, and I will hand over to you to introduce yourself to everyone on the line and dive into your presentation.
Thank you, Jennifer, first of all, for having me here. So my name is Vaibhav Narayan. I'm currently at Johnson & Johnson, working on various initiatives related to digital health, especially in the mental health and dementia space. And I'm happy to speak to you about some work that we did recently over in Europe as part of our IMI project called RADAR CNS. And I think I'll go through these slides relatively quickly, and hopefully we can reserve most of the time for Q&A. So RADAR CNS is an IMI-funded public-private partnership co-led by Janssen and King's College London, Professor Matthew Hotoff, which, as you can see from the screen, is in fact a fairly substantial collaborative partnership with many participants, both from, like I said, the academic side and as well as the industry side. And essentially the goal, RADAR, in fact, stands for Remote Assessment of Disease and Relapse. And we started with CNS. And we are studying three conditions here: major depression, multiple sclerosis, and epilepsy as well. And so the work that I'll describe today essentially is a byproduct of this work that we're doing in RADAR CNS, where what we have done is we have reused some of the data that we were already generating in these patients, and reuse that data to understand the impact of COVID on these patients, since these studies happened to run at a time when COVID hit the countries we were operating in. So obviously a big thanks to IMI for their funding right at the very beginning. Next slide, please. So as part of the RADAR CNS consortium, what we have done is developed essentially an open source platform called RADAR Base. And RADAR Base platform is the platform that we are using to essentially collect all the digital phenotyping data RADAR Base is, like I mentioned, an open source, extendable plug and play platform, which can be used to collect both passive data from devices as well as active data from self-reports, etc. And on this slide, you can see the various sort of types of data streams that we handle in RADAR Base. And also, I should mention that RADAR Base creates a dashboard where you can monitor at a population level the various data streams that are coming in. And in fact, I had presented this slide at FNIH Biomarkers Consortium, and sort of and reusing that slide, which is always a great thing to do. I should also mention that RADAR Base has its own website, radarbase.org, and I encourage you to go there and get more information about this really cutting edge, nice open source platform that we have developed to enable digital phenotyping studies in various settings, including academic settings, research settings, and other settings. And there are many aspects to this platform that we believe fill a very nice niche in this space. And so I'd be very happy for you to sort of dig into this more, and the various developers that are developing this platform would be, I'm sure, happy to be contacted by you. I should also mention that RADAR Base is already being reused outside the RADAR CNS consortium in various research cohorts across Europe. So it's already gaining both a nice sort of acceptance in different research studies, and also has a fairly active open source development community around it. So I definitely encourage you to go to the website and also follow up on this platform later on. So next slide, please. I should also sort of, I threw in the slide again more as a plug. So as I mentioned to you earlier, in RADAR CNS, we are essentially conducting digital phenotyping studies in MDD patients, in MS patients, as well as in epilepsy patients. And these are long-term studies. We are following these patients for up to a year or 2 years. In MDD patients, we have about MDD cohort, we have about 600 patients. We have a similar number in the MS cohort, and epilepsy is designed a little bit differently. And I guess on this slide, I just wanted to make the plug that before we started these digital phenotyping studies, we spend a fair amount of time understanding how we would pick the devices and the technologies that we want to incorporate in these studies. Okay, and we have actually published this work, human-centered design strategies for device selection and health programs, and also patients measurement priorities for remote measuring technologies. You can see both these references at the bottom. The crux of the matter is that we wanted to design digital phenotyping studies where patients will be engaged for the long haul. We are collecting a lot of data. We are throwing the kitchen sink at these folks, frankly, because we see these as signal finding studies. And these are non-interventional studies. So nobody was getting feedback directly about the key clinical conditions we were following. So the question was that how would we have an engaged population? So we took a very patient-centric view. We not only explored the technological landscape, but we had patient advisory committees. We really understood from patients what their motivations were for participating in these types of programs. And based on that, we selected the right sets of technologies and devices. And so I do want to make a plug for this work also. It's very important as we do digital programs to really keep the patient at the center of your design. And again, if anybody in the audience has follow-up questions around this, we would be happy to sort of share our learnings in terms of how to keep all of this very, very patient-centric. Next slide, please. So now, with that said, right, we get to the aim of this the current publication that we are talking about, and essentially at a high level, what we wanted to understand was that what was the utility of a platform like RADAR Base, which we had deployed in these patient populations, in assessing the response to a COVID lockdown. And I think I think that ultimately, the broader context here is that if there are any non-pharmaceutical interventions that are given at a public health level, at a population level, can we use a platform such as radar base and data from devices and smartphones to really understand how individuals are responding to such interventions, and so as I said, fortuitously almost, the fact that we were doing this digital phenotyping studies already gave us an excellent opportunity to test this hypothesis. And since these studies had started actually way before COVID hit, we had the benefit of having some nice baseline data as well, right? And this, I think, is actually an important point to make, because most of the other kind of studies that have come out have essentially looked at cross-sectional kind of readouts, just at the time of COVID or post-COVID, and very few had the advantage of having longitudinal data for almost, in the case of in UK, for example, for almost a year before COVID hit. And so this was something definitely which we think is an important part of this work. So next slide, please. So like I said, the data were already collected from 5 European countries: UK, the Netherlands, Italy, and Denmark, from November 2017 onwards. In some cases, not in all countries did it go that far out. And we had a total N of 1,062 participants, and they all have either MDD, major depressive disorder, or multiple sclerosis, because it was essentially data from these two studies that we reused. We did not include any data from epilepsy patients, because actually our epilepsy study was an in-hospital study. But as you can see, these 5 countries were the countries that were most severely hit by COVID. So it did really set up this experiment nicely. And we went back to our funders and said, listen, our primary goal, of course, is to understand relapse and disability and all of that in MDD and MS, and that's what it's set up for. But would it be okay if we, as a consortium, took a look at COVID impacts, et cetera? And of course, they said yes. This is taxpayers' money after all, and they were happy to have these data be reused in this context. Next slide. So again, I think I'll go relatively quickly through this. I'm assuming you guys have read the paper. We extracted features from smartphone and wearables in essentially 3 categories: mobility, physiological measures, and phone use. We compared behavioral differences for the baseline, pre, and during lockdown phases, right? And the baseline here is, of course, the many months or almost a year that we had before there was any hint of COVID in the air. Pre is just before the lockdown was announced, and during lockdown is, of course, when the lockdown was in place. And we also looked at some other cuts related to age, gender, BMI, and educational background, and I'll touch upon that briefly. Next slide, please. So with on the next slide, I guess all I have is a list of the extracted features. So we were able to get a sort of a privacy-proof version of a location, maximum distance traveled from home, homestay, etc. You can sort of read in the paper some nuances about how we define the home location based on the location, where they last sort of, shut the phone down, or their last sort of touch with the phone. We were able to also look at how many times is the smartphone interacting with nearby Bluetooth devices. In some ways, that gave us an idea of their social distancing measures. We had a Fitbit step count as well. So between the location, the number of handshakes, and the step count, we thought we were able to get a reasonable sort of measures of mobility. For physiological measures, we relied mostly on Fitbit data, bedtime, sleep duration, light, deep, and REM sleep, and Fitbit heart rate, the average heart rate, daily average heart rate. And of course, we had phone use data as well, the unlock duration, how many times you unlock the phone, how long did you keep it unlocked, as well as we had data on how much time people were spending on various social apps, and we used the Google Play categorization to track these the usage of different apps here. Next slide. So very quickly, I'll just go through some of the high level conclusions, and I think I want to spend some time talking about limitations and next steps here, because while this is a nice first publication, what we think is perhaps even more relevant is what are the other things we can do going forward. So hopefully we'll get to that as well. So if you just ignore all these, Jennifer, just click through all these click-throughs. So yes, we found some things that you might expect, that as lockdowns sort of hit, people obviously spent more time at home. This was from the location information. They traveled less. They had fewer nearby Bluetooth-enabled devices. That they were more socially active. Socially distanced and physically distanced, so to speak, but they were not socially distanced, I would say. So they were more active on their phone, in particular, interacting with others using social apps. So yes, there was physical distancing, but that does not necessarily translate to social isolation and social distancing in this day and age of social media availability. Next slide, please. Again, like I said, just please click through, Jennifer. I apologize for this. We also found that participants had lower heart rates, which might be because people were sleeping more as well, and they were also going to bed a little bit later. Everybody's schedules were obviously a little bit off, with children staying at home, etc. But we also found that while people were sleeping more during the lockdown, in some countries we found that people actually slept significantly less in the days leading up to the lockdown announcement, where the initial sort of COVID had spiked and there was uncertainty about what's going to happen next. And there's sort of an interesting observation, which has implications for some of our future analyses, that just before the lockdown, in fact, is perhaps the more sort of anxious and more stressful times, when there is no certainty about what's going to happen. And so if we want to sort of look at the impact, for example, of COVID on things like depression, stress, and anxiety, it's not simple. You cannot simply do that by comparing just before lockdown to lockdown. You need to have long-term longitudinal data, because the pre-lockdown phase itself is fairly perturbed, we think. And in fact, in some cases, lockdowns, we hypothesize, may even have resolved the uncertainty, and people went back to a nice routine, et cetera, et cetera. So things are always a little bit more complicated than you might imagine. But anyway, this was the high level sort of readout of what we saw. Next slide. Keep going. We also saw different responses across countries, right? We did split our 1,000+ cohort into the different nations and took a look at this. We could argue whether that's a good split to do or not, but we did do that. And what we found, and you can see this in the paper, that there is sort of a clear change that you can see in most behavioral and physiological measures in most countries based on baseline to post-lockdown. But Denmark showed reduced responses, attenuated responses. They were not that, the signal that we had in Denmark was definitely lower. And it was interesting to note that Denmark was the one country that had a different sort of approach to the lockdown. They had strict restriction on workplaces and public transports, but were much less strict on social events. and homestay. And so this sort of public policy could have created the difference that we saw in terms of physiological and behavioral changes. Now, I'm not suggesting that you can use these data to say that a certain type of lockdown is more effective or not, but I guess I am suggesting that you can use these data to it, to understand if certain types of lockdowns are less effective or not, depending on how you want to define effectiveness. But we can certainly see that the nature of lockdown did sort of reflect in our data in terms of the impact it had on the behavior and the physiology. Next slide. Keep going. So like I said, we also did some other cuts. We found that younger people actually stayed at home longer and walked less compared to older adults post lockdown, which may be a little bit counterintuitive, but if you think that the lockdown was thou shalt not leave our house but for critical crucial activities, turns out maybe younger people are more savvy about using online services, whether it's grocery shopping, etc., and they were much more effectively able to hold themselves at home, whereas maybe older folks didn't figure out how to use these online services and still had to go out and get some essential work done. By the way, our definition of younger, and some of you may react negatively to this, is less than 45 and over 45, and so that it's just an interesting signal we found, just sort of, and it may be, like I said, to do with their ability to use online services. We also found that people with lower BMI remain more active during lockdown, um, versus people with high BMI. Again, we can speculate maybe people with low BMI already had home gyms set up and treadmills set up, and they were able to continue to sort of indulge in those activities. Those educated to a degree level stayed at home for significantly longer in Italy and Denmark. And this may be because people with higher education maybe had jobs that they could do from home more easily, and they didn't have to get out. And I would say that I say Italy and Denmark, but many of these measures trended towards significance in other countries as well. So while we call out two countries for statistical significance, does not necessarily mean that this was something unique to these countries. Next slide. Keep going. So again, I guess this is like a high level conclusion that we think that we can extract these kind of features from smartphones and wearables using the radar based platform, and we can look at the impact at baseline, pre and post lockdown for COVID in these individuals. And we did demonstrate the utility of such platforms in monitoring physical and social behavioral changes. On the next slide, if you go, Jennifer, I do want to obviously point out some of the limitations of this study. Next slide, please. Hey, Jennifer, is it just me, or is the slide not moving forward? Right. So some obvious limitations. Obviously, this is a specialized population. It's in MDD and MS patients, so that might affect the generalizability of some of these results. Having said that, in one of the countries, we kind of looked at the to see if there were obvious differences between MDD patients and MS patients, and we found relatively few or insignificant differences. It is a limited N, right? It's 1,000 plus. It is not one of those massive sort of contact tracing studies that Google and Apple are able to do. On the other hand, this is a study where we have longitudinal tracking of patients or individuals or participants over a long period of time. We are able to collect information from them through self-reports or specific questionnaires. We are able to follow through around certain outcomes, etc. And so there are many things about this study that also make it unique, even though it's a smaller N. It's like I said, more dense tracking, and we have actually a two-way data type with the individuals, and I'll come to that later in terms of some of the next steps. And of course, another limitation is participants were recruited at different times and seasons in different countries. We tried to address this a little bit. So for the baseline period, we went back to the individuals sort of data from the similar season, sort of in the last year, so that we were able to counteract seasonal effects. But we were not able to do that very effectively in all countries where some of the studies were started later. But this does reflect the real world, and we just have to learn how to handle these data than to sort of get paralyzed by these limitations, I would say. Next slide, please. So future work, right? So like I said, this is not just a sensing study. We are actually able to interact with these individuals and push questionnaires and collect data. And so one of the things that we are doing is actually sending COVID questionnaires to all the participants in these studies to really understand, okay, the lockdown happened, but did they actually get COVID symptoms, et cetera? So that's another aspect that we are pursuing, and we will see how many of these patients actually had COVID-like symptoms or in fact did get COVID as well. And an obvious thing to do for us is, of course, since we are tracking things like depression, anxiety, and stress, that's part of the design of the study. In fact, that's the fundamental goal of this study. We have various outcome measures related to mental health, both through self-reports as well as through digital biomarkers. So the next obvious thing to do is to understand the impact of COVID lockdown and even COVID symptoms on the mental health, both digital and self-reported outcomes. And the other thing, of course, is that can we use the longitudinal data almost in a predictive setting, right? Could we have said that, okay, people who, for example, developed sort of COVID symptoms had certain changes in their respiration rates, in their sound and speech quality, upper respiratory sort of acoustics, in a way that we could have picked up the onset a little bit earlier than we did. I was reading this interesting article that, in fact, there are AI algorithms being designed to detect COVID cough, which might actually be inaudible to otherwise. And so I should just let you know that we are collecting speech and voice data and et cetera in this study as well. And so there is the potential of doing some predictive modeling. Of course, the challenge is that, thankfully, from a patient's perspective, we hope that not a lot of these individuals end up getting COVID symptoms or COVID, and so that will limit sort of the predictive modeling kind of capabilities. But I think we can still look at predictive models of, say, symptoms that are related to COVID-like illnesses, and so that's something also that we are looking to do. And I think I'll stop here. I mean, I try to give you a sense of what the study is, what the data are, and would be happy to take questions.
Vaibhav, that was terrific. You walked us through not just the study itself, but I think all of the important components that were in place in order to allow you to ask these sorts of questions. You're also my favorite person today because apparently I'm a younger adult. So that's terrific. Also, thank you for your patience with me with the slides. We have a hurricane blowing into town, and I saw a trash can fly past my window, so had to deal with that for a minute. And I'm delighted to say we already have some questions coming in. Silvina, I am going to unmute you from my side. If you would like to pose your question directly, please go ahead. And if you, terrific, you should be on mute.
Hi, yes, wonderful presentation, Vaibhav. Thank you so much, very interesting. So my question actually, partially you have already answered, but I would appreciate if you could give more details about the next use cases that you can envision for this type of digital phenotyping platform from the RADAR CNS?
Yeah, so I think, Selena, thanks for the question. I think our primary goals of the digital phenotyping studies are in fact related to understanding and quantifying disease scores and predicting relapses in major depression. And so that will be on track. In MS, we want to really understand disability and fatigue in MS patients, and also understand relapse and drivers of relapse in MS patients. And in epilepsy, we want to develop a regime that will allow us to both detect and predict oncoming seizures in these patients. I just wanted to point out that those are the primary goals of these studies in MS, MDD, and epilepsy, and those are very much on track. And we are actually collecting some pretty remarkable data. I mean, we have had terrific engagement, terrific adherence in these studies, and I believe are collecting unprecedented longitudinal data sets in these cohorts. We have, for example, observed more than 100 seizures in our epilepsy study already, with all the data being collected. The MDD study is fully recruited with more than 600 patients, 620 patients, followed, like I said, between a year or 2 years. Likewise for our MS study. So that will continue. In terms of COVID, I think that our next goal is to understand the impact of these lockdowns on the mental health and the behavioral sort of mental health conditions, because remember, these patients already have MDD, right? They already have major depression. So it's a good place to find out how lockdowns and even post lockdown scenarios impacted their mental health, not just right then and there, but over time, right? Were there changes in outcomes and trajectories, etc. So we are nicely set up to ask some longer term questions of the impact of COVID and lockdowns on the mental health sort of trajectories of these patients. And that's I think going to be one of the very next steps. We also have a paper that we are in the process of publishing, which is going to look more closely at the impact of these lockdowns on sleep in itself, right? And so I think we'll be just digging deeper into those types of questions.
Silvina, that was a terrific question. Really appreciate you asking. Mark, I'm going to come across to you next. I've unmuted you from my side. You should be able to unmute yourself and ask your question directly.
Yes, thanks for that. So I really kind of appreciate this presentation. It's very, very interesting, and kind of relates very closely to what we in my company are kind of focusing on around MDD. I'm particularly interested whether you've been able to uncover any insights around kind of the relationship of your sample size. As you were mentioning, it's still relatively limited, but I'm particularly interested about the relationship between the sample size and what you've been able to see in accuracy of predicting things such as relapses. If that question makes sense.
It does make sense. Let me just start by saying that, our view sort of going in is that when it comes to predicting relapses, there are two things. There is the depth and the breadth of the data, right? I think more important than the N of the study is the time, the longitudinal sort of data collection, right? How much data do you have for an individual over a period of time, so that they can serve as their own baseline, and we can essentially develop personalized predictive models on these individuals. So we will be looking. So it may not be a general population level model that you can apply on everybody, but the model would be sort of what we call an N of 1 model potentially. But I cannot answer your question directly because we haven't yet formally started the predictive modeling on relapse, although we have in the MDD set more than 100 relapses that we have already recorded based on the definition we had of a relapse going in. So to those who are active in the MDD space would realize that this is actually a pretty substantial accomplishment, because it takes time, following somebody for months or even years to encounter a relapse, right? And so we do have the data set. We are first answering some more lower level questions before we start developing the predictive model of relapse itself. So I cannot answer your question, what the impact of N on that model is going to be, but I can say that we are looking, we will be looking very closely at at the longitudinal nature of our data more than anything else. And I think that's where the most of the power is going to be, not the fact that we followed, a few hundred patients, but the fact that we followed them for a long period of time.
Thanks, that was very helpful. Thanks.
Mark, thanks for a terrific question following hot on the heels of Sylvina's. Excuse me. Sandy, you've also put a terrific question into the chat. I've unmuted you from my side. You should be able to unmute yourself and pose your question directly if you'd like to.
Yes, thank you. Hi, Vaibhav. Thank you very much for the presentation. I used to work in biosecurity, so I was very intrigued by the article. And you mentioned that fortuitously you had this baseline data on the group of people that you were following. If you were to imagine, like in real life, and hopefully this wouldn't happen, but in a future pandemic, would different countries essentially need a baseline established for mobility for different geographies or regions within their country and different populations in order to be able to understand the effectiveness of any NPIs put into place? And did you and your fellow researchers explore really sort of the privacy concerns around any of that?
Thanks, Sandy, for that question. Yes, I think, ours is obviously a consented study going through an IRB, and we have sort of various ethics committees. And so we have all the right privacy provisions in place, which we take very seriously. And in fact, it's because of privacy concerns, our data are treated in a way where we kind of address those issues. And so therefore in the location, we cannot tell exactly where folks are, right? Obviously, not just, at a local level, but even more globally. And so therefore we cannot say which regions of, Spain, for example, had less mobility or more mobility. All that is somewhat obfuscated. And I think that so I think in this case, we imagine, everybody's reference was the North Pole, right? And we're just looking at mobility around the North Pole. We have no idea where exactly you are. On the other hand, I do believe that as patients get more control over their own data, over time, people can choose to sort of have a baseline established for themselves, right? And there could be ways of then looking at deviations from that baseline in a way that are meaningful and helpful, both at an individual level and then de-identified at a population level as well. And I think the trick here is that how do we kind of get individuals to consent to this kind of use of the data that they are generating? And I think one very important way of getting them to do that is to show the utility of these types of analyses, both at an individual level, where we can, for example, predict and track if something untoward is happening to your baseline, and also at a population level where we can get some useful insights. So people are motivated by being citizen scientists and helping contribute data to a population level effort that helps public health authorities, et cetera. So there is an altruism here as well. But you're right. I mean, for that to happen, folks would have to be aware and willing for certain types of data to be collected and tracked over the long term. And like I said, I think these data already exist. We have to proactively, prospectively, and transparently get consent from individuals to reuse these data in the ways we are talking about.
Great, thank you.
So for me, I think the big key word is data reuse. That's the only sustainable way of doing it, rather than brand new data collection. Everybody who has a smartphone and a Fitbit already generates these data,
right?
More or less.
Okay, thank you.
Sandy, thank you for a terrific question. I see, Akash, that you have a question. I'm going to come to you next, and I'm going to be absolutely vile and dive in and follow up on your question, actually, Sandy, around consent. What was the consent process for the parent study, Vaibhav? Did that allow you to reuse the data, as I love that language, to answer these questions around the impact of lockdown without going back to the participants, or did you have to reconsent?
I don't want to misspeak on this one, so I'll get back to you whether we reconsented or whether our original consent allowed this or not, if you don't mind. Yep.
Yeah, of course. The reason I ask is, my mind goes to sort of this idea that, presumably, right? There is an opportunity, because this is sort of government funded, for folks to come in perhaps through RADAR base and sort of use these data later. So yeah, I'd love to know if you wouldn't mind sort of following up there.
Yeah. Yeah, we could. I can say that our original consent was, pretty comprehensive, but this one I just want to double check before I say in a recorded forum that we did this. But for example, we have consent from these individuals to be able to connect back to their EHRs and things like that. So the study is nicely set up for increasing the richness of these data in various different dimensions.
Fantastic, appreciate it. Akash, over to you to pose your 2 questions, actually.
Hi, Vaibhav. Thanks for the presentation. Very helpful. I'll stick to one of my questions just to respect everybody else. So in terms of the future work that you were discussing, being able to quantify the impact of depression, anxiety, and stress, what is the motivation behind trying to collect digital biomarkers for this data versus something a little bit more traditional, like validated questionnaires, ePro, et cetera?
Yeah, well, I guess, quote unquote, depends on what you mean by validated. I think one high level issue in mental health is that there is no sort of ultimate gold standard, is there, right? But to answer your question, yes, we are collecting psychometrically valid sort of questionnaires as well, right? And so that's not, it's not an either or. We are collecting a lot of validated, established scales. But one of the questions that we are asking in these studies is that in addition to these scales, right, can we collect data passively in a low friction way, which would also help us quantify these disease conditions and perhaps give us a predictive signal that may be more sensitive and easier to collect in a large scale than repeatedly sending out questionnaires. But we have various questionnaires, both as sort of input measures, also as outcome measures. We also have EMA, like ecological moment reassessments, where we give a burst of questions repeatedly, multiple times a day for a week or so. So there's a pretty rich data collection. In fact, I would encourage you to look at our publication. We have published the protocol of this study, RADAR MDD. I should have probably put that in somewhere, and you will see all the data that we are collecting. And I believe that that should answer a lot of these questions that you have, Akash.
Cool. Thank you very much.
Akash, thank you so much. Terrific question. And Vaibhav, let's plan to, if you want to shoot me that link, I can make sure that that goes into the slides before we post. I don't see any other hands raised, so I'm going to pose a question of my own. One of the things that really impressed me about your data from this study is the extraordinary engagement that you've been able to maintain over a period of time that positioned you not just to achieve sort of the original goals of RADAR CNS, but to pivot and to ask these COVID questions because you had that strong baseline. Can you tell us a little bit more? You had that lovely slide around sort of getting it right and starting with the patient and being incredibly deliberate. But there's a lot of technologies, a lot of them are active. How have you made this successful? And what would you recommend to other folks on the line who are trying to do these digital studies that typically have horrible drop off?
Yeah, I mean, I think it's a question that even we are asking ourselves, honestly, because I think we have even surprised ourselves in terms of the level of engagement and adherence that we have had. And part of it, you could argue that, well, it's a self-selected population, right? I mean, these are people who agreed to participate in a study like this. But even so, we looked at that, and about half the people we had approached declined to participate, half of them did, and that's fine. But even so, I think it's pretty good adherence. Part of it, I have to sort of acknowledge the research team that we have. So these are not completely uncared for folks, right? If somebody has trouble, they can sort of, call somebody and people follow up with them. So there's a level of engagement, which I fully appreciate is not sustainable in the big, broad world that is happening in these studies, that is creating, sort of an, high level of engagement and adherence. But like I said, to offset that, I would say we are really burdening these patients with a lot of data collection, right? Lots of different streams, lots of apps on their phones, various, Akash was asking about self-report questionnaires, ecological movement reassessments, wearing a Fitbit. And in the MS studies, in fact, there are other devices that they are wearing as well. So yes, it's a high touch study in some ways. On the other hand, it's also high data footprint, right? So in the future, we hope to reduce the touch, but we also hope to reduce the amount of data that we collect and then and the burden on them. So hopefully that will make it sustainable in the real world as well, because from these types of studies, we will understand what is the minimum data that you can collect for the maximum value. The other thing I would say, Jennifer, again, is that our understanding is that clearly articulating the research goals of the study to the participants and clearly articulating how these data would help, their physician or to take better care of them. I think those, at least my view is, those are the kind of things that motivated these individuals to stay on. And there is also an element of, we don't share people's depression data or clinical data back with them, but we do share some summary metrics and things like that. And what we are finding is that, people with depression and people with MS are actually fairly interested and curious about some of these data that we are collecting, and they are happy to sort of continue to stay engaged with technology. So I don't want to sound like we have somehow cracked the problem of long-term engagement and drop off. No, I'm sure we haven't. These are studies where we are paying a lot of attention and we are doing a lot of follow-ups. So we are very sort of realistic about this. But on the other hand, we hope to publish this soon. We have remarkably high retention and engagement rates.
You really do, and thank you for sharing some of that with us. It's fascinating, and frankly, I feel like there's been so much published around these. You just see these precipitous drops. It is possible. That's what I take from this, right? It is possible with that sort of good communication with participants, really articulating that value prop, to be able to have this kind of, prolonged engagement. So congratulations to you and the team.
And one thing I would add, Jennifer, to that is that COVID might be a little bit of a game changer going forward in people's acceptance and willingness to participate in remote assessment studies. And in fact, in some ways, these studies, the fact that we continue to keep the study going, very nicely through all the lockdowns and COVID-related sort of issues, just points to the fact that in fact, to the strength of the fact that we are able to do remote assessments, first of all, and secondly, our hope is that people now will be much more willing to adopt technology, going forward. But again, like I said, at the end of the day, I don't want to underestimate the challenges as well, especially if you go into a kind of a well population. I think it might actually be more challenging to keep people engaged when you are perfectly fine and you don't have any diagnosed illness and you don't have a physician in the loop and you don't feel like somebody is necessarily looking at these data and making it actionable, right? The fact that we are in a population where they have a clinical diagnosis, where they understand that we are trying to do things for them that have not been done before and things like, I think all that is sort of helping in this case, at least.
Fantastic. Those are really interesting insights. And actually, Claire, I'm going to invite you to pose your question because I think it's a really lovely follow on here. You should be able to unmute yourself and pose it directly to Vaibhav, Claire.
Great. Thank you all so much. This is an awesome cohort and publication. Really looking forward to some of the subsequent work that it sounds like you have underway. One thing following on Jen's question, and that I do think is a real opportunity potentially that's come out of COVID, and I've been asking it of all of the studies that had these kind of COVID moments where the lights went out at the Super Bowl, if you will. How did you communicate with the participants in the study as the pandemic set in? I think particularly with this study about how the research was evolving. And do you think that impacted your adherence, in this case, potentially enhanced your adherence, that you were in touch more than others? And how do we extrapolate that more broadly to clinical trials?
I think actually it's a very good question, and we will take this back and see how the sort of the increased engagement and the fact that we are sending out COVID questionnaires, etc. How did that sort of impact the overall adherence to the study, and did it go up? So Claire, thank you for posing that question, and we would hopefully, when some time passes, be able to actually answer that question. But to your broader point, for example, we were collecting speech and voice samples, and we recently reached out to all our participants to sort of explain to them about why we are collecting the speech and voice data, what digital biomarkers we expect to extract, and so on and so forth. And now we'd be putting our ear to the ground and seeing whether that communication increases the adherence to these speech and voice tasks, because we are actually collecting sort of specific tasks, right? Count to 1 to 10, describe a happy event, right? It's actually, people have to actually sit down and record their voices. So is sort of a communication around that task helpful in increasing engagement around that task, or is it communication around COVID increasing overall adherence? I think we will learn a fair bit about motivations and adherence. We give people exit interviews. We have all sorts of built-in mechanisms to get patient feedback. So I don't have an answer right now, but I'll put this down as a question for the team.
Great, thank you.
Fantastic, Claire. Thanks for the terrific question. Sandy, you posed one of my favorite questions in the Q&A. Go ahead and ask your question directly to Vaibhav.
Sure. I want to go back to the issue of data reuse. If we were to imagine a longitudinal study with a cohort of, say, 40,000 people where you are reusing their different device data, how much does it matter whether the devices are consumer grade versus medical grade? And then how do you account for the differences amongst devices in a very large study?
Oh, well, that's like the hit parade of key questions, isn't it? But let me go back to some, I guess, standard answers, but I get the question behind your question. The standard answer, of course, whether it should be consumer grade or medical grade depends on the intended use, right, and the risk of such use, etc. If you are predicting sort of an event that which has implications in terms of clinical decision making or things like that, then that regulatory framework sort of becomes tougher and tougher, and it's no difference here. Although, having said that, I would say that at least in RADAR, what we have said is that our goal is to use as much as possible consumer grade devices, right? And then really reuse that information. In this case, of course, we are just trying to see what the signal there is and how, what regulatory path does the signal have to go through, and what implications does that have on the device hardware itself in terms of, is it medical grade or not? I think those are questions that we will have to answer in the, going forward. But I expect the data reuse to happen, hopefully as much as possible from whatever devices the individuals happen to have on their wrists, right? So it's a bring your own device regime. We have to carefully track the intended use and the risks associated with that, I would say. In terms of device independence, that's also a good question. We have some other studies that we have done internally and otherwise as well. And I would say that the more we can get access to raw data, the easier it is to solve the problem of device sort of dependence, right? It is when you rely on summarized and processed data that's where the device dependence becomes even more acute, because now it depends on what algorithm was used to sort of make, extract these features and do this processing. The more we can develop our predictive algorithms or digital biomarkers directly on the raw data, the better, the more likelihood we have technically of being device independent. But then of course the devices have to make the raw data available, right? And so then that's another set of questions over there. But these are hopefully, questions that Jennifer, you from your forum can sort of, get towards, right? How do people, different device manufacturers, get to some sort of data standards? Can we have data contracts, APIs with certain guarantees on certain, parameters, and ensuring certain amount of data quality? Can we get access to raw data? I don't think that, any one consortium or any other study is going to solve that problem. But I think if we can show the utility, at least, of this data reuse, then there is that push to solve these problems.
Great, thank you.
Fantastic, Sandy. That really was. I love your description there, Vaibhav. The sort of the hit parade of tough questions about technologies. And it's funny that you mentioned that this is right in our wheelhouse, because we are doing further work on our playbook to advance this. And also we do have a new tour of duty really planned on trying to think about how to use particularly sort of consumer technologies and how to keep them consistent and communicating with each other over time for exactly these sorts of data sets. That will be starting next year. So Vaibhav, what a tremendous conversation. Thank you for being willing to join us in the hot seat. You did an absolutely marvelous job today, and you covered a lot of ground, not just the terrific publication on COVID-19, but you also briefed all of us on the tremendous work of your colleagues on the work group over at RADAR CNS. So thank you very much. Thank you also to folks on the line for joining us. These are really terrific discussions. I appreciate how engaged you all are with the conversation, and I have a couple of pages of chicken scratch here thanks to your good questions. Quick look ahead for folks who enjoy these smaller groups. It's certainly one of my favorite forums. Our December, I didn't update that, I'm sorry. Our December virtual journal club is going to look at a really terrific publication in Health Affairs that starts to think about how, when we use these technologies in patient care, that we actually go ahead and get reimbursed for them. What is the perfect model there? So Vaibhav, you set the bar high today. We've got another terrific discussion coming up next month. Thank you everyone for your time today, and have a terrific afternoon.
Thank you, folks.
Thank
you, Jennifer.
Of course. Thank you. Bye-bye.